Thanks for being here. It's great to be able to start with prayer at the beginning of a talk. Alan said we're going to pray because we're BYU and we can. I like that. I mean, we're now the Ability Lab and we can't really. But we pray a lot, we just don't do it in front of talks. But I like that a lot. Greetings from Chicago. We can talk a little bit about our new brand. We used to be the Rehabilitation Institute of Chicago, and we just built a new $1.2 million science medicine laboratory called the Ability Lab, which is disguised as a rehabilitation hospital. And our goal is to make patients better, which has always been my goal. And even when I was in San Diego, I was working in areas that were applied, and I had the fortune to work with some great people. I just thought I'd feature 4 of them today. Dave Sutherland is an orthopedic surgeon who introduced me to the field of cerebral palsy. And since I was a muscle guy, he said, you know, there's all these muscle things, well, you should study them. But, I didn't tell him I was a frog muscle guy, and I assumed there were some big differences between frogs and kids. And so I sort of laid in the background, but over the years, our lab transformed from an animal lab to now it's only humans. Jan Fridén is a longtime collaborator. He's a hand surgeon in Sweden. I'll show you some of his work. Hank Chambers is a pediatric orthopedist and sports medicine guy in San Diego, and you'll see some stuff we do where we get muscle biopsies from the operating room from some of these kids. And these are typically Hank's patients. And then Lucas Smith is a PhD student who did a lot of the work that I'll tell you about today, and he's now at Penn. So let's go ahead. So I like to start with this picture because I like this multi-scale view of muscle. And I also use it as a little bit of a disclaimer, because when you take your classes, everybody teaches you, it's like malpractice if you don't learn about actin and myosin, right? In fact, one of my students' 2 dogs are named Actin and Myosin, which is kind of cute. I like that kind of commitment in a student. I don't think she was just doing it to butter me up either. I think she loves proteins too. So we learn about actin and myosin, and we learn about myosin being the molecular motor that powers muscle contraction. In fact, myosin is the molecular motor that powers lots of stuff in the body. And one of the reasons I love studying muscle is the kinds of things you learn in muscle are then often applicable to other systems. For example, calcium signaling in muscle. Calcium is the switch that turns on muscle contraction. Turns out calcium is activated in a lot of biological systems. I already told you about myosin. ATP was discovered in muscle. So a lot of things that apply across biology are most obvious in muscle because it's got such a specialized function. So in the foreground, we see the sarcomere, the business end of a muscle, the contractile machinery, if you will. A sarcomere is arranged in series into myofibrils, and these are surrounded by a pretty sophisticated membrane system. So here's another example. The so-called sarcoplasmic reticulum, which is responsible for releasing calcium, activating muscle, and then taking it back up, is a specialized form of the endoplasmic reticulum. And if you study biology, you know that trafficking in cells is a big deal. So one of the areas this has been learned is in the muscle world. And then as you get to the background, you see muscle fibers, which are giant multinucleated cells. That becomes real important because while they are multinucleated, in between the cells are a bunch of other things that are important. Extracellular matrix composed of capillaries, nerves, other stem cell population, fibroblasts, connective tissue. And in the background is where a lot of the important questions of muscle remain to be solved. And probably as exercise scientists, that's the area where you care most. But it's not the area where physiologists have necessarily done the best job, and a lot of it is just really the lack of tools. So I'm going to talk to you about a little bit of tool development today as well. Now, I told you about the sarcomere. The most significant functional property of the sarcomere is that the length of the sarcomere predicts its force. So on the horizontal axis here is sarcomere length in microns. So these are pretty small. A micron is a millionth of a meter. So just think about that for a second. Muscles move large distances. Giant muscles like mine, like these biceps muscles. It's not supposed to make you laugh. You're supposed to go, yeah, that's right. No, I'm just kidding. These biceps muscles can move centimeters, but each sarcomere can only move microns. So that's a lot of movement going on in series. It's like when you're walking on the escalator. Right? The escalator's moving at speed, you're walking at speed, and your speed relative to the ground is faster. Same thing happens here. The summed movement of these sarcomeres is huge, sometimes centimeters. And the important take-home lesson that we teach in physiology is that the force is length dependent. So these, the pink line is supposed to be myosin, the white line is supposed to be actin, and you can see that muscles have an optimal length. And it's it's obviously a very important feature. When the muscles are too long or too short, they don't work as well. So it's interesting, we teach this in physiology, and then a smart student might go, okay, well, what is the length of muscles in the body? And we usually say, generic teachers say, well, pretty much this optimal. And I'm just here to tell you today that's turns out to be almost never true. But my PhD, I told you, was in frog muscle. And one of the guys on my committee was a comparative morphologist, and he said to me, why should muscle have a length dependence? Why is that a good idea from a design point of view? And, I was totally stumped. I was just a sarcomere guy. I was so into sarcomeres. As far as I'm concerned, I was barely even married. I just loved sarcomeres. But I figured it was a fair question. And it was the kind of thing that it didn't really haunt me, but it bothered me. And I started studying the way sarcomeres move in animals. In other words, what's the physiological operating range of a sarcomere? And since at the time, Andrew Huxley was the guy who was defining sarcomere lengths and how they generate force in frogs, this became our model system. So here's Iraноpipians, and we took the frog apart and left the semitendinosus muscle in there and could measure the detailed properties of the muscle, the tendon, and the amount of torque it generates by just stimulating it in vitro and hooking it up to a force transducer. And this little jig here would allow us to rotate the hip joint or the knee joint or whatever, but we'd be able to get the relationship between sarcomeres and these joint angles. And this was a big lesson for us. This was work that Jenny Boakes did. Jenny was a medical student at the time and is now an orthopedic surgeon in Sacramento. But the vertical axis is sarcomere length, and the horizontal axis is joint angle. And if you remember your sarcomere lengths, happens to be knee joint angle, with the knee fully extended, the sarcomeres basically are generating almost zero force. And with the knee fully flexed, they're generating near maximum force. And I shouldn't tell you this story, but Andrew Huxley, he was my hero. Sliding filament theory, Hodgkin-Huxley model of action potentials, Nobel Prize, not too shabby. He had told me, we know that sarcomeres operated at their optimum. And and there's a lot of implications of that. And even at the time when he told me that, I thought, oh shoot, I didn't know we knew that. And when you're a student, you think some old guy tells you something, it's probably true. Turns out it wasn't true. Like none of these sarcomeres are near their optimal. And so what that tells me is their length dependence is kind of built into the system. And in this particular system, since it was all definable, this is how it ends up looking. So this is this is the force generated by the muscle as a function of knee joint angle. That's because its sarcomeres length change length. This is the moment arm, the mechanical advantage at the knee joint. It's pretty much a hinge in a frog. And the important thing is, here's this yellow line is the joint moment, the torque generated by the muscle acting at the joint. And I think one of the most important lessons here, and it's really important if you're studying humans, is that strength, which is measuring joint moment as a function of, say, joint angle, is a complicated interaction between muscles and joints. And for example, in the frog, if you have a frog clinic and you're testing frog strength, and frogs are strongest at 110 degrees of knee flexion, okay? Why? Well, it's actually kind of a boring answer. The boring answer is it's just kind of the mathematical sum of the muscle force and the joint torque where it's optimizing. Force is going up, moment arm or mechanical advantage is changing, and so it's just the optimal solution to muscle force and joint torque. So when you're lifting weights and you know you have an optimal joint angle, that joint angle doesn't necessarily mean it's the angle where your muscles are strongest. In fact, like I said, in the muscles we've measured, it's not. And if your goal is to strengthen muscles, the way you strengthen a muscle is you apply stress across the fiber. And really, if you were training frogs for the Frog Olympics, you would train them with the knees more or less fully flexed. That's the angle where you get the most muscle force. Your torque is low, but that's just a low mechanical advantage. And I work in a rehab hospital now. I tell our therapists and our physiologists, we have exercise physiologists, that those kind of parameters are critical for patients. I mean, you could make a patient 25% stronger by just exercising them at the right joint angle. That's an important point. And of course, it has lots of clinical implications. So that's sort of a basic science view of why sarcomeres are important in vivo. Clinically, why they're important is as a surgeon, a lot of times the job is to reconstruct, let's say, a wrist or a hand. And with my buddy Jan Frieden, we were very interested in studying the decisions surgeons make during reconstructive hand surgery. So we developed a tool, basically a fancy laser pointer, that we could slide into a muscle. And I told you already that muscles have these sarcomeres. The sarcomeres have an optical banding pattern, the so-called A bands and the I bands. They refract muscle and they produce a diffraction pattern, just like in basic physics. It's a periodic structure. You have a wavelength of light that you can use. And so you can use these devices in the operating room in real time to measure sarcomere length as you're moving joints around. So this is an example of isolating a bundle of muscle fibers, dissecting down to a usable level, putting the little laser device underneath the bundles, and then turning it on and measuring sarcomere length. It's kind of like having a microscope on the patient on the operating room table, right? Because you get a number in microns. You can sit there with a laptop and just dial in the patient to their exact sarcomere length. Super cool. Like our patients are 10 times stronger than the normal surgery because we get them at the right sarcomere lengths. That's cool. So what do you find when you measure those sarcomere lengths? Well, I'll show you a couple of examples to just tell you what it means. In this one example, this is the wrist extensor carpi radialis brevis. The left-hand side of the screen is the wrist flexed. The right-hand side is the wrist extended. And you can see here's a situation where the wrist, when it's fully extended, the extensors are generating their maximum force. Now just think, if you're God, you can talk about God at BYU, which is really nice. If you're God and you're designing wrists, and you're using sarcomeres and the wrist is its maximum force potential with the wrist extended, what would you do about flexors? Where would you put the flexors? And how would you change that as you rotate the joints? And why is that a good idea? So that's 10 years of work that I'll summarize, but I just wanted to sort of plant the question in you. And it turns out it's kind of non-intuitive. So this is how it works out. These are the 5, as you know, if you know your anatomy, there's 5 prime movers of the wrist, 3 extensors, and 2 flexors, if you don't include palmaris longus. The wrist flexors tend to operate at short sarcomere lengths. The wrist extensors tend to operate at longer sarcomere lengths. So with the wrist flexed, the flexors are short and the extensors are long, and both of them are generating less than optimal force. And as you go from flexion to extension, the flexors become longer and stronger, the extensors become shorter and stronger, and the net result is a really nice balance of torque throughout the range of motion. And if you know hand function, the purpose of the wrist and positioning the wrist is really designed to be a stable base for finger manipulation. And if, let's say, we put flexors and extensors at their optimum with the wrist in the neutral position, you'd flop off on one side or the other in terms of strength as you moved your wrist and during normal everyday movements. So we would say the human wrist joint, kind of the musculoskeletal design, is a balanced design. And this has a lot of implications. So if you talk about sort of evolutionary significance of mammalian musculoskeletal design, a lot of times people, when they're looking for evolutionary selection pressures, they say, well, you're optimizing power production or you're optimizing force production. One of the implications of this design is you're not giving the joint the biggest bang for the buck. This joint is not designed to produce the most torque. If it were, you would always have maximum force in the position where you had the maximum mechanical advantage, and you don't. This is a balanced design. Now, the selection advantage to that might be, better dexterity, better ability to eat, survive. But I will say that so far what we've learned in terms of musculoskeletal design is we find very little human systems that are really designed for peak power production or optimal force production. If you're a biomechanical modeler, a lot of times people talk about constraints in modeling. The systems are complicated, so you always have to superimpose some constraint to solve your equations. And sometimes the constraints are for optimal force production or peak power production or optimal efficiency. I just don't see that as a human trait. I just don't see us doing that in terms of musculoskeletal design. And we can talk about that more. Now, we were doing this work in Gothenburg, Sweden by this time. Jan had moved down to southern Sweden. Right next door to us was a operating room where they were operating on kids with wrist flexion contractures, which I had never seen. So here's about a 10-year-old kid. He's got bilateral wrist flexion contractures. In fact, his contractures are so bad that his wrist joints are dislocated. So he has cerebral palsy, which is a perinatal brain injury, either during birth or right before birth or somewhere around birth. There's a brain injury, and then these kids grow up, and as they grow, sometimes, depending on the kind of treatment they're getting or not getting, they end up with muscle contractures. Wrist joint muscle contractures are very common. Knee flexion muscle contractures are very common. Hip adduction muscle contractures are common. So kids with these contractures, whatever the strong side of the joint is, will typically win. It's analogous to people, you might see people who've had strokes who end up with elbow flexion, wrist flexion, shoulder internal rotation contractures, so their limb will be held like this. That's because those are the strong sides of the joints, and they can affect locomotion as well. So we knew this was going on, and the treatment for a contracture, actually, if it's gotten to be this bad, the treatment is simple. These are typically, as you probably know, the flexor carpi ulnaris is the strongest wrist flexor. You literally go in and just cut the tendon, and the muscle retracts like a rubber band, and then the kid can at least move their wrist a little bit with residual function in their extensors. So it's not a super fulfilling surgery, but it's fairly common. So we were doing our work in spinal cord injured patients and tennis elbow patients, which is another story. These children are being operated on next door, and the chair of the department says to us, you really ought to study these kids with CP. And I'm a regular old scientist. I'm a simple man. I watch football, I drink whatever, Diet Coke, I don't know, I do simple things. I run. So the idea of cerebral palsy, muscle contractures, you gotta be kidding. I mean, what is going to brain? I don't even know where the brain is. I know it's up here somewhere, but I've told you all I know now about brains. So I'm thinking, we don't want to do this. So she said, yeah, I understand. And plus we had lasers and reconfigured the operating room, and we were training nurses. So she said, we really like that you're here doing your science in Gothenburg. We love that you're willing to help us with our patients, but you're actually taking a fair amount of our resources. And I am in charge, she's talking, I am in charge of scheduling the operating rooms, and you are taking extra time, and as the chair of the department, I really think that you studying children with cerebral palsy is a good idea. And I go, I've always wanted to. It's a great idea, great idea. And I'm not kidding, it wasn't too far from that. So we go in there, and I just look at Jan just go in, shut up, stick the laser in, blah, 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 and we'll go out and we'll do our stuff. Right, okay. So the good news is it was a totally Swedish-speaking OR staff next door. And we had our little laser tool. So we go in, and here's the shocking result. I should actually have some kind of picture of the event itself, but I was all scrubbed. Here's the crazy result. The kids with wrist flexion contractures had sarcomeres in their wrists that were super long, super long. And in the diffraction world, long sarcomeres means the diffraction orders are very close together and super bright. So typically, we'd stick the laser in the arm, it's kind of dim, you turn off the OR lights, you look, you measure, just like I showed you in that movie. These were razor sharp and super bright. And I instantly knew, because I'm a nerd, that meant long sarcomeres, highly regulated. It's oh my gosh. And I looked at him, and we both just were in shock, because it was such an amazing result, which is a good scientific lesson, right? You don't think you want to do it, it's a little tangential, give it a try. What's the big deal? It takes a half hour of your time, or whatever, maybe a little bit more. Give it a shot. It's a chance to do something very different. And this turned out to be a really huge finding, because it's unprecedented. There's there's lots of muscle diseases out there. There's no long sarcomere muscle diseases until this. So and how does this happen? And I'll tell you, based on other studies, we knew sarcomere length was highly regulated. So this doesn't just happen because there's less activity in the muscle or something like that. And if you go back to your length tension curve, you realize that these long sarcomeres have very significant functional implications. And we know our kids with cerebral palsy are weaker than normal, and we know that they have smaller muscles than normal. And we always thought they were weak mostly because the muscles were small, but now you can tell from this study that they're weak in large part because they're at the wrong sarcomere length too. That's a big deal. So what's going on globally? Well, sarcomere, that's just one little measurement. Maybe the rest of the muscle is normal, who knows? So we started doing a bunch of other stuff. I'll just show you one example. This is work that actually Margie Mathewson did. This is the soleus muscle of a child with cerebral palsy. Margie was using ultrasound to measure the length of the fascicles in the muscle, and then wheel those kids into the operating room and measure their sarcomere length. So if you know how long the fascicles are, and you know the length of the individual units of the sarcomeres, you can calculate how many sarcomeres are in series. That's really what determines the range over which a muscle will operate. That's a hugely important functional parameter that we don't know very much about in the muscle world. So here are her data. The fascicle lengths, you can see cerebral palsy versus typically developing. The typically developing kids were kids having ACL reconstruction or had tibial fractures where we could get to the soleus muscles. And the sarcomere lengths, like I showed you before in the wrist flexors, were much longer in the soleus muscles of kids with CP, sorry, compared to typically developing. And what that means is, if the fascicle length is the same and the sarcomere length is about half, there's about twice the number of sarcomeres in series in a typically developing kid versus a kid who's got cerebral palsy. That's a big deal. That has huge functional implications. One is the weakness I told you about. Another one is when you stretch sarcomeres, they're under lots of tension. So these contractures, in part, there's a little bit of fibrosis going on in the tissue, but these contractures are in part due to these extremely long sarcomeres, which put the child at a huge disadvantage. Now, you know, when you do something like this, you're assuming that you understand the building blocks of the muscle, like the sarcomeres and all that jazz. So I'll tell you, we did spend a lot of time. I was born and raised in the single fiber mechanics world. We did spend a lot of time measuring the fiber properties and the tissue properties. So here's a picture. It's a really neat assay. It's like super high tech. It's one of those things that's kind of disappointing, like we're measuring a single cell from a human being and doing physiological testing on it. Really? Yeah. Well, what'd you find? It's normal. That's the letdown. But it was super cool experiments to where we could find out at the cellular level that the force generating machinery was typical, was normal. What was interesting was when you went to fibers or bundles of fibers, which maybe is 10 or 20 fibers, that was not normal. Their mechanical properties were altered, and the biggest thing that was altered was the passive mechanical properties of them. So if you look at like a histological section from kids, on the left-hand panel is typically developing kids, on the right-hand panel, kids with CP. This happens to be a stain for laminin, but it would look the same if you were staining collagen or fibronectin or any kind of extracellular matrix protein. And you can see, I'm Jewish and we have Yiddish words. There's a lot of schmutz here, okay? The schmutz here is stuff that we don't really know about. And it was, again, surprising. You see it in other conditions. You see it in muscular dystrophy. You see it during muscle regeneration. You can see it with prolonged spaceflight, disuse, denervation, stuff like that. But remember, the nerve and the muscle in these kids have not been affected. It's their brain that's affected. And you'll have to trust me on this. It goes way beyond what you see with disuse atrophy. So we started, this is mid-2000s, we really started studying the extracellular matrix of muscle. And I had been born and raised in an orthopedic surgery department, and I thought, okay, well, extracellular matrix, that's like tendon and ligament. These guys know all about tendon and ligament. Somebody must have done this in muscle. There's very little done in muscle. And I say this a lot to physical therapists and other clinicians. When you're feeling muscle or doing a physical exam on a patient and looking at things like range of motion, et cetera, you're feeling mostly connective tissue. I'm convinced of that now. We can talk about it. I used to think you might be feeling other muscle properties, but I can tell you why I don't think that anymore. So we just started measuring things like measure the modulus of the bundle, like a normalized stiffness, in either semitendinosus or gracilis, these are hamstring muscles, and measure collagen content. And kind of lo and behold, the collagen content is up and the stiffness is up. It's probably a collagen issue, and now we have to understand how collagen is organized in muscle. And I put this little picture on there to remind myself, this is a classic temptation that I've got. I measured this, it goes up. I measured that, that goes up. And as you know, correlation isn't the same as causation. But it even turns out when you take these exact specimens and you regress the modulus on the collagen content, you can see it's kind of a shotgun blast. There's really not much of a correlation. So the kids with CP have more collagen, but the amount of collagen they have that's more isn't really what's giving them the greater stiffness. And that led us to think that it's really the structure of this extracellular matrix. Like if you took a Volkswagen and a Mercedes and you boiled them both and ran them on a gel, you'd get a bunch of steel, a bunch of rubber. Yeah, in one case you'd have genuine Corinthian leather, in the other case you'd have vinyl. But that's different, but a lot of it would be the same. So composition isn't a big deal necessarily, structure is big. So I did what any good professor would do. I talked a new student into studying muscle extracellular matrix, and I'll just tell you one of our tricks. I'm a little bit out of the business now, so now I'll tell you my trick. One of the good tricks of being a professor is bribing a student to do something that you don't know much about. In this case, it was Allison Gillies, and then calling a friend and saying, there isn't really much on the extracellular matrix. How about if you invite me to write a review on the topic? So then you write an invited review on the structure of the extracellular matrix for Muscle and Nerve, which is, it's an okay journal in our field. And people think you know about it. Well, you know about it because you just read something and you know a little bit more than the next person did, like a month before they learned it. But you look like an expert. So it's a really good strategy to look like an expert. Write a review. And it's even better if you have a friend to get you to invite the review and they label it invited, because like why else would they invite Liber to write the review? You must know a lot about what he's talking about. It's not true. But I did it anyway, and I've done it many times, and I will continue to do it, but I'm sharing it with you. Okay, so what do we find? Two things. One is the basal lamina of a muscle cell, which is mostly type 4 collagen. It's hard to get pictures like this. We did a lot of this at the scanning electron micrograph level, which isn't great. If you know the method, scanning electron microscopy requires drying the tissue and carbon coating it and stuff like that, and things shrink, as George says. There's shrinkage here for sure. In fact, the reason that it's so important is I showed this to a guy who does microscopy, and he said, "This is total BS." He goes, "How'd you prepare the sample?" And I told him all this stuff, and he goes, "You have no idea what's going on in that system." So I'll tell you what we did for that guy. But anyway, turns out he's right. We got fooled, and we found some stuff by just God's grace. The first thing we found was that the collagen arrangement right around the muscle fibers, the way you get this kind of image is you dry the tissue and you literally kind of, it's like if you took 2 soaking wet rolls of toilet paper and let them dry, and let's say you glued the ends together of the 2 rolls, and then you put them in water, let them dry. Now you try to unroll the rolls and get toilet paper, right? It flakes off, it just breaks. But every so often you get lucky and you'll get a piece that kind of goes out in the middle between them. That's what this guy is right there. And you can do structural analysis on it. Turns out it's boring. It's like a net. So it looks like a woman's nylon stocking or the things at the grocery store that hold oranges. It's a structure that just goes in whichever direction it needs to keep the volume constant. So I was telling my wife this. I thought this was really interesting, like female, helpful stuff. I'm at the little stand with the legs, and I'm looking at large, medium, small, or however they're rated. I go, this based on this structure, it really isn't like tall, short. All it's doing is doing a volume constraint. It's like, it should be called like large volume, medium volume. Anyway, not very well appreciated, but right. And my wife is small volume, so that's good. So there was that. Boring structure. The very interesting structure was the other stuff. Out in the paramecial space where there's bundles of fibers are these long cables. And we don't teach our students about paramecial cables. I didn't know about them. Of course, they're in the literature from the 40s and 50s, but we forgot about them. And there are a lot of these huge cables in the periphery and tons of collagen fibrils. And this is where my buddy, he's a brain guy. The neuroscientists are always smarter than regular muscle people. He developed a 3-dimensional reconstruction method for the cerebellum to do using electron microscopy. We stole the methods and applied them to muscle, and this is part of Allison's thesis, where here's a muscle fiber, here's a muscle fiber, here's a muscle fiber. Here is a little piece of a cable, but you can see it just looks kind of like a smear. Here's a capillary, here's a capillary, here's a big bundle, here's like an isolated cable, and these are the 3-dimensional reconstructions. And these are the movies that you get from this kind of thing. This is 170 nanometer slice at a time going through hundreds and hundreds of microns of muscle tissue, and then reconstructing them into these movies that are on the right. So here's a piece of a cable. Actually, down here is a piece of a fibroblast. This blue guy over here is a fibroblast. The fibroblasts in muscles are huge. What this gives us is quantitative in situ values for all of the ultrastructural parameters that in that one picture I showed you with schmutz just looks like goo. And we can sort out what it is in the extracellular matrix. We can do it biochemically, we can do it morphologically. And to make a long story short, the difference between a stiff muscle and a compliant muscle, or in this case we'll say a kid with CP and a typically developing kid, is the fraction of collagen that ends up put into bundles. That's what's shown here. This is really painstaking stereology done by a student, Rachel Meza, who's now in PT school in Chicago, where we did this kind of stuff and quantitatively laid out grids. And so the image I have in my brain now about how muscles regulate passive stiffness is there's a certain amount of collagen in them, but there's somebody in there who's putting collagens into bundles that are load-bearing and just having collagen on the side. And collagen is a little tricky to assemble and a little tricky to synthesize. So I think the logic there is it makes muscles able to respond a little quicker, but it's pretty expensive. I mean, muscles keep a lot of collagen around that aren't necessarily ending up in structural properties. And I think this is a brand new area where we're all going to have to be doing some serious work. Okay, so at this point, my grant is just about out. I'm trying to think about renewals. And I know I have long sarcomeres, I have weird extracellular matrix and all this kind of stuff. Started talking to friends about, you know, how are these things regulated biologically? And that plus some other sort of fortuitous things led us into the whole area of trying to understand the stem cell population of muscles. And as you probably know, muscles have satellite cells. Look at this. Oh, an invited review. Oh, that's kind of neat. So obviously I didn't know very much about that, which is why I invited myself to write a review. And the guy bit. He's a very nice guy. You probably know muscles are a cool tissue. And I mean, I could give a lecture called The Top 10 Reasons Why Muscles Are Cool to Study and why if you want to be cool, you should study them too. But I only do that with my own students. But one of the reasons it's cool is muscles have a built-in stem cell population. And it lasts pretty much your whole life. And that's what we refer to as these satellite cells. And here's another cool fun fact, boys and girls. Satellite cells were the first stem cell discovered based on morphology. There was a little cell in the corner of a muscle fiber, and they were, I mean, this is in the 60s, viewed as some very unique cell type, and they were called satellite cell because they were on the periphery of the muscle fiber. Since then, you know, people have studied them many other different ways. But you can see here's a picture of one that's on top of the cell, and then the basal lamina, the collagen coating, would be right on top of that. So that's shown here schematically. This is a review by Sudarshan Dayanidhi, who's a physical therapist, PhD. So here's the basal lamina on top of the cell, and there's this idea that satellite cells basically sit there quiescent, like many stem cells, and they only spring into action when bad stuff happens, like muscle injury. Eccentric exercise is a really good way to turn them on. Snake venom is a good way to turn them on. Any kind of trauma is a good way to turn them on. They're used during development. It's a really interesting process. If you watch how muscles develop, remember I told you they were multinucleated? The cells, cells divide. They are multinucleated. They start fusing together, but they don't all fuse. Some of them stay behind. And the ones that stay behind end up being satellite cells. Nobody knows exactly why some of them stay behind, but that's another theme in the stem cell world, asymmetric cell division. That's a big deal in the stem cell world. Because if stem cells just make more of themselves exactly, they'll all do exactly the same thing. They don't do exactly the same thing. They do mostly the same things. That's a big thing, has tons of clinical implications. There's a bunch of satellite cell biotech companies out there. So you should keep your eyes open for that. So the reason they're important is when muscles are injured, they turn on and they recapitulate development. Or when muscles need to grow, like on January 1st when you decide this is the year you're going to finally get in shape, and you start lifting weights and you get bigger, you're getting bigger because your muscle fibers are getting bigger, and your fibers are getting bigger because they're adding satellite cells. They're recruiting satellite cells to them. And that's a normal physiological process. Not really very well understood at the molecular level, but it's clear that it happens. So we thought maybe there might be a satellite cell involvement in these contractures. We're not talking about muscle fibers getting fatter, we're talking about muscle fibers getting longer in these kids. And there's evidence in the literature that fibers, in order to get longer, also recruit satellite cells. So we did kind of a crummy study with Lucas, where we just did cell sorting of muscle after the single cells had been isolated. And you can see here, this is the satellite cells, endothelial cells that line blood vessels, and inflammatory cells, which are just sort of floating around tissue within muscles. Kids with CP in the blue and typically developing in white. And you can see there's a huge drop-off of satellite cells in kids with CP, which is a really important finding, because think about it. If satellite cells are required for regeneration and very, very helpful, although the molecular literature wouldn't say required, but super important for growing muscle, maybe the reason these kids can't grow their muscles is they just don't have the right number of satellite cells. That would be a big deal. And there's nothing a physical therapist is going to do to make them work, right? If they're intrinsically defective as a muscle, they're just not going to be able to grow. It has all kinds of implications for repairing them, growing them, regenerating them, and even therapeutic interventions. So we thought, okay, this was, I was at this time, I was in business school, deciding that I was going to build the world's best hospital. And in business school, you always have to do a project. And I figured my project's going to be, I'm going to grow satellite cells in kids with CP, grow them up, inject them back into the muscles, and cure their contractures. Wouldn't that be cool? A non-surgical treatment for muscle contractures. Take a muscle biopsy from the wrist, take it to the lab, grow a zillion cells, inject them all over the place, get them back into the cells, do PT, voila, all better. That was the idea. It was a good idea. So the step one is grow them. And so we started growing satellite cells and started looking at their properties in kids with cerebral palsy versus typically developing kids. And we found they move pretty well. You can see no significant differences in terms of movement early on after we harvested. They proliferate very well. Division, in fact, they proliferate even a little better than typically developing kids. What they don't do is they don't mature. So on the vertical axis here is myotube area in percent. So you can see probably from these pictures, here's typically developing cells down here, here's CP cells. They're kind of spindly. They might have a couple of nuclei. You can measure the number of nuclei per myotube. It's higher in typically developing kids versus CP. So this is another really important finding for two reasons. One is it means that the company that we started to do this, to fix this problem is dead. Really fast. So that was fun while it lasted. We didn't even have business cards. It was fun while it lasted. That's important. But the other thing that's important about it is it means that this whole brain injury that happened and led to, the problems, contractures, et cetera, somehow altered the stem cell population. Like there's something else going on with these cells. And actually that sort of turns out to be good news in a way, because there are lots of ways to modify stem cells. The most common are all the various epigenetic imprints that happen on cells, which you've probably studied. I mean, epigenetics is a great new field, and one of the best parts about it is there's huge numbers of epigenetic modifications in cancer biology. So there are FDA-approved drugs for epigenetic modifications out there already that we can potentially test on our muscles and our kids and our cells. So if you look at the cells themselves, this happens to be, again, just more cell culture, typically developing kids versus kids with CP, same amount of time, same juice. We really thought if we took the CP cells that weren't developing well in situ and put them in cell culture medium, which has all the growth factors and all the goodies, they'd grow great, but they just don't grow and they don't mature. And where we are now with these kids is starting to understand the molecular basis for the lack of development. And the classic pathways that you would look at are developmental pathways, such as focal adhesion pathways, right? When cells develop, they grab onto a substrate, and if the substrate's mushy, you probably know this, if it's mushy, they don't develop into the right stuff. Like bone cells need stiff matrices, muscle cells need compliant matrices. Or if they can't grab, like they don't have the right fingers or the beta integrin signals in muscle. Here you can see that the kids with CP are expressing a lot less of the beta-1 integrin than typically developing kids. And we're looking at the epigenetic imprints that are causing those changes. And so in the end, we've ended up with a system that we can study at the cellular level, at the single fiber level, sort of at the tissue level, and at the in situ level. And we're headed now toward screening drugs using this system and being able to use those drugs on our kids. It's a really interesting thing, right? We have a very small patient population. We've been talking about it for the last 2 days. The medical system is messed up. So you can make a lot of money studying muscular dystrophy because it's a so-called orphan disease. And there's lots of biotech companies that study muscular dystrophy. There's almost no companies that study cerebral palsy, even though it's the most common childhood reason for mobility problems. But it's not an orphan disease. And the other thing is, treating it doesn't make you a lot of money. So if you're going to treat kids with cerebral palsy, you're going to have to do it in a clever way. We have teamed up with people who understand the FDA and understand biomedical companies to screen existing approved phase 1 FDA drugs. So phase 1 means it doesn't hurt anybody. The first thing you ever do with a drug study in people is you feed it to them and you see if they get hurt. If they don't, yay. Phase 2 is, does it work, okay? So there's, for every 100 phase 1 drugs that pass, there's 5 phase 2 drugs that pass. So 95% of the phase 1 approved drugs, and phase 1 approval probably costs between $5 and $30 million, which we don't have, but they're sitting on the shelves of drug companies. So we've been going around to drug companies, and you can talk to a drug company about a specific biological pathway, and they'll say, oh yeah, we have a focal adhesion kinase inhibitor, yeah, Well, can we try it? Sure. So we actually have screened, in the last couple months, we've screened a bunch of drugs, and we actually have some drugs that'll completely reverse this, which is really cool. I can't show you the data because we need to publish it. It's submitted. I'll send you a paper individually, but I'm not willing to get it videotaped. The cool thing about that is, suppose it works on the cells, which it does. I mean, literally, by law, a physician with IRB approval could give it to a kid tomorrow. It's already an FDA-approved drug, not for CP, but for cancer. And potentially, we could treat the kids, give them this pill, epigenetically remodify their satellite cells, then give them physical therapy and all the stretching and all the kind of stuff you do, and then maybe reverse these contractures. Because right now, our treatments, which are stretching, splinting, casting, Botox, et cetera, don't work very well. They work okay, they don't work great. But this is a way to kind of go after a biological solution to these contractures. So, to put it all together, oh, no, I'm going to tell you one more thing, sorry. I told you this. I told you that there's not many satellite cells in the kids with CP, and I told you that their serial sarcomere number is down. They have contractures. And those are two observations, and the question is, are these causally related? And so in San Diego, I had a big mouse lab, and we have kept a couple of our mouse models. There's a way to test this idea, right? We borrowed a mouse from John McCarthy and Charlotte Peterson. Charlotte Simmons. That's a book, right? Charlotte Peterson. Where we can get rid of satellite cells. It's a really cool model. You just take a mouse that's been genetically engineered to express diphtheria toxin, which kills cells, every time the PAX7 promoter is turned on. PAX7 is the transcription factor that makes satellite cells. And it's inducible. So you can take a mouse, he's living his life, he's having a happy time. You can feed him tamoxifen, and you can kill selectively satellite cells in his muscles. That's a pretty cool model, right? So we can test the idea, does satellite cells cause contractures? And what we did is you can see here, we're immobilizing mice in plantar flexion, which shortens their soleus muscle. When that happens, the soleus gets shorter. Soleus muscles don't like to be short. Sarcomeres don't like to be short. So it gets rid of them in series, sort of like a contracture. Then we feed them tamoxifen, get rid of their satellite cells, and then we let them run around and see what happens. And here's kind of the cool result. The sarcomere number with vehicle-treated soleus muscles is about the same, But our casted guys, even after 4 weeks of walking around, their sarcomere number is still down, and they still have a plantar flexion contracture, which is pretty cool. So this is, a mechanistic demonstration that satellite cells are also involved in that serial sarcomere number addition thing. And it kind of, it's obviously it's a mouse, and it's not cerebral palsy. It's a satellite cell depletion model. It's not the same thing, but it is very interesting. Okay, and the last thing I'm going to show you is kind of a fun little project that's new, because we're really interested in developing tools for humans. And I showed you laser diffraction, but I also showed you, I saw a few of you wince. It's pretty gross to cut open people, and it makes it really hard to do experiments. What do we need to know sarcomere length. What are you going to do? Well, we're going to cut you open. Like there's only a few idiots who will let you do that. But we do have a new tool, and I'm just showing you it here. We just published this thing, and it's a cool, basically fiber optic thing that we can slide into a muscle. And the trick here is that we use, I have a collaborator who's kind of a telecommunications guy. We have a lot of ways to take multiple wavelengths into the muscle. The multiple wavelengths can diffract from a muscle at any sarcomere length. Remember I showed you n lambda equals d sine theta? So when you have a fixed wavelength, you get a band at a fixed angle. When you have every wavelength, you get a band at every angle for every sarcomere length. And with the right detectors, you can measure any sarcomere length you want. It's a very cool method. And so not only that, you don't have to just measure sarcomeres. You basically can do kind of in vivo spectroscopy and look at metabolites. Molecules have all these signatures, and we're starting to measure these kind of compounds in muscle. I was just going to show you the data from, this is a rabbit muscle, but you can see it's a pretty cool experiment where the red line up here is sarcomere length. This is a single twitch of a rabbit. Down here is the spectrum as a function of wavelength, and down here is another optical thing we don't need to talk about. And you can see the noise in the system is a fraction of a nanometer. For just one little twitch, we can easily measure it with just a little fiber optic probe. We're starting to do this in kids who are walking around. And we can actually do it during surgery when a lot of surgery is less invasive now, so you just expose the distal tendons and you can make specific measurements on distal tendons. So we're really hoping that we can do this for sure. We know we can do it fiber optically. Our DARPA grant is to do it through the skin. So we're kind of developing one of those Star Wars things where we can scan the skin and get sarcomere lengths. That's for us kind of one of the next frontiers. Okay, so what have I told you? Muscle contracture represents a complex and I would say uniquely human muscle adaptation. So mouse, cat, rat, and rabbit muscles do things that human muscles don't do. There are no contractures. Even when you do head injuries and spinal cord injuries in these animals, it's not exactly clear why, but there isn't really an animal model of contracture. The passive muscle mechanical properties are altered to a greater extent than the active mechanical properties, and that's bad news because muscle physiologists and everything you learn in school, I mean, how many people here took a muscle course? Be honest. A lot? Should be a lot. Okay, shy people. Anyway, mostly we teach you about active muscle mechanical properties, not passive. Because we don't know that much, and we teach what we try to teach what we know a lot about. The CP muscles have trouble growing. Whether it's radially, I didn't show you the data, but fibers are always smaller. And longitudinally, for sure, I did show you the data where the sarcomere number is lower. They have trouble growing. We think that's a satellite cell issue. But all of our therapies are hampered because we don't really have knowledge in several specific areas, which is the topic of all my new grants. Extracellular matrix structure and function, the regulation of that structure and function. Remember I talked to you about like isolated collagen fibrils, how do they get into bundles? Who's in charge of that? And then the whole satellite cell development and function, that's not my area. It's a huge area. If you do a literature search on that, you'll find 1,000 papers this year on it. But it's a big deal, at least in CP. So we're being forced to study it. So I just acknowledge the funding agencies who pay for this stuff, and I'd be happy to take questions or hear your comments. Thank you.